Next, we welcome David Weinstock of Dana-Farber Cancer Institute and Scott Manless of MIT. Together, they share a grant defining vulnerabilities of minimal residual disease. Please welcome David and Scott.
Great. Matthias, do you have the... super. Okay, great. So thank you very much. It's a wonderful honor to speak again with Scott. We have multiple disclosures. The one that's most relevant is Travera, a company we co-founded that utilizes the SMR technology developed by Scott. So our focus has really been on trying to validate a hypothesis. And that's based on the fact that we put most patients with lymphoma into complete remissions using standard chemoimmunotherapy, but many, many patients relapse. And so the concept is we treat them until they're in a minimal residual disease state, and then we either hope they're cured and stop therapy, or in some contexts we continue therapy, and then they break through with resistance. And so our hypothesis really was that if we had the technology capable of isolating and then characterizing minimal residual disease in an iterative fashion, that we could target therapeutics toward vulnerabilities within that MRD, and then potentially even cure patients if we could ultimately eradicate the MRD. Presumably, it feeds through a limited number of pathways. Now, there are potential advantages of targeting MRD as opposed to treating patients when they have full-blown disease. Most notably, there's likely to be less clonal complexity, and the patients are likely to be in a better performance status than when they have full-blown disease. The obvious disadvantages are that the hypothesis may be wrong, and we may be giving patients therapy that's of little or no benefit to them. So we started by trying to pilot this concept within patient-derived xenografts. The technology that I'm going to mention and then Scott's going to discuss further involves a microfluidic device to very precisely characterize biophysical properties of individual cells, most notably the cellular mass. And this was particularly attractive to us because it's a measure that's independent of immunophenotype. And we know that there's a significant amount of plasticity within both lymphoma cells and precursor cells that are not fully malignant, but harbor a set of mutations and can serve as a reservoir for secondary transformation events. So a patient may have one type of lymphoma and then develop a second type of lymphoma. And so we orchestrated trials within these patient-derived xenografts of BCR-ABL rearranged leukemias. So this is a large number of mice xenografted with a large number of different leukemias and then treated with tyrosine kinase inhibitors. And the advantage of this system is that we can induce very deep remissions for a prolonged period of time, 150 to 200 days. And we perform serial bone marrow aspirations of these mice to isolate very, very rare cells. When the mice relapse, they commonly harbor either reactivating mutations of ABL-STAT signaling or orthogonal mutations, usually involving MAP kinase that activate similar pathways. But it's really within the MRD context where I think the data is most interesting. We can now take these very rare MRD cells and we can perform paired biophysical assays and single-cell RNA-seq. This is in collaboration with Alex Shalik at the Reagan Institute, using primarily his seqWell platform, but also other platforms. And then we can try to identify different vulnerabilities at the time of MRD. And because these xenograft models are quite pliable, we can then return back to the model and determine whether combinations that are nominated through this analysis can actually eradicate disease. And it turns out that in some contexts they can. I'm just going to show one bit of biophysical data. And this is actually pair data where individual cells are matched both for their biophysical properties, their mass and their stiffness, as Scott will discuss, and also their single-cell RNA-seq profiles. And it was quite illuminating to us. So what we notice is that the mass distribution of cells, this is prior to treatment when the mouse has hundreds of millions of leukemia cells, you see follow a distribution that almost looks like a cell cycle analysis, where you have these cells that look like they're in G0 and these cells that are in G2. When the cells get to MRD, so this is tens to over 100 days into a minimal residual disease state, now we see that the cells seem to have arrested, or at least they're smaller. But in fact, what they're enriched for are programs of, I apologize, a more differentiated state of B cell. So you can see that here. And in fact, that state continues to predominate when the cells now relapse. So they're actively cycling, they're overwhelming the mouse, and they are actually a more differentiated progenitor B cell. And we surmised through immunophenotypic analysis and transcriptional analysis that we could then target that either with Syk inhibitors that block this pre-B cell receptor signaling or with inhibitors of P38 MAP kinase. And in fact, in combination now with TKIs in the same models, we can essentially eradicate all disease in a very short treatment period. So I think this is proof of principle that the concept is sound and it could now be applied to patients with lymphoma. But there remain three major challenges. The first is capturing the heterogeneity of minimal residual disease within a human being. And really, in order to do that, we think we need to sample multiple sites. That's extraordinarily difficult in a living human being. The second is that we need to be able to isolate and characterize extremely rare MRD cells, and to some extent that involves increasing the throughput through our process. And the third is, to go back to the point I made, that any biased isolation of minimal residual disease based on an immunophenotype leaves out the possibility that there are cells that have a different immunophenotype that are actually serving as the reservoirs of relapse and or secondary transformation events. So we need immunophenotype independent identification methods. So to get at the issue of sampling multiple sites, we've taken a number of approaches. We've set up a pipeline to be able to get every biopsy of lymphoma performed at Dana-Farber, for example. But we've also developed a rapid autopsy program. And I think this is a tremendously powerful approach. We were admonished at the last Allen meeting that in Paul's memory, what we should really do is hit it out of the park. And I think that the trainees who are actually doing this work deserve an enormous amount of credit because it seems like all of our rapid autopsies happen between 2 and 8 in the morning. And so they're up all night isolating individual cells. The patients and families really also make a remarkable sacrifice. So not only does the patient obviously sign up for an autopsy that's performed sometimes in less than an hour after death, but that means that the family is also not able to sit with the patient after they die. So they're really making a sacrifice for this to happen. This is an example of a patient who had a T-cell lymphoma that was metastatic to the brain. And for those of you who are used to looking at brains in section, you'll see an expansion of the white matter. The patient was treated with high-dose methotrexate and then had further inflammation and succumbed to the disease. Had a large immune infiltrate, and we thought that the disease most likely had progressed. But actually, as we went on and looked at this immunophenotypically and transcriptionally, what we learned, in fact, was the patient was in a complete remission. They simply had a massive inflammatory response within the brain in the setting of the therapy. And as you can see, all of the cells in this t-SNE plot, other than this very small cluster here, are microglia. And they cluster separately based on, I've only shown 2 locations. We performed 12 based on the location within the brain. And so these microglia have different signatures based on the geography and the location of the disease. Even within the T-cell cluster, you can see most of these cells are actually CD8s. So about 1 in 300 cells was actually a malignant cell. But we can easily pull those out based on bioinformatic algorithms that call copy number variants based on single-cell RNA-seq data. We're sequencing deeply enough. So we can identify MRD at a rate of 1 in 300 from multiple different sites, and we have this paired transcriptional and biophysical data. And so Scott, I think, is going to take over now and talk about the approaches that we've taken to get at the other limitations.
Okay, thanks. Okay, so just David mentioned a lot already in terms of the rationale of looking at the biophysical properties. I'll just say a little bit more. We know through the eyes of pathologists that there are changes in cellular structure with disease. This is 2 examples from my colleague Dino De Carlo showing both that the shape and the size of cells can change as disease is tracked. But there are some limitations for what a pathologist would be able to see through this. One, as David mentioned, if the cells are rare, that becomes very difficult to do this by imaging. Not impossible, but difficult. 2, there is an inherent bias in that the pathologist is looking for particular types of known patterns to relate to this. And then 3, there could be structural changes in the cell that just may not be resolved by the type of microscopy that's readily accessible to do this type of work. And so this is what we would like to try to address using the biophysical readouts. So for cell size, we're going to measure cell mass. It's not the same as size, but it of course is related. In cell shape, we're thinking about the cortex of the cell and the mechanical properties. Again, not the same as the shape, but something that has some relations. So let me just briefly show you how we're making these measurements. So in terms of mass, we have this microfluidic device. It's in the shape of a diving board. It's vibrating at a resonant frequency. As a cell flows through it, the frequency changes in proportion to how much the cell weighs. Just like if you were on the end of a diving board, it'll be vibrating more slowly than when you jump off. And so that little frequency response tells us the mass of the cell. And just to give you an idea of the precision, if we take the same cell and weigh it as it's growing, so this is a lymphoblast, it's a cell line, doubles in 10 hours, we're just flowing it back and forth through this device and subsequently weighing it over a 30-minute period that you see here. So just to calibrate you in terms of the scale, so one picogram here is about a 75 nanometer equivalent change in diameter, but our resolution is around less than 50 femtograms in terms of mass, and that's like the cell were to change its diameter by 5 nanometers. So these are changes that are very hard to see in other ways, but we can readily resolve them through this method. So that's mass. Let me say a few words about the stiffness. So the cortex is the outermost part of the cell, mainly comprised of actin, filaments, and myosin, and to a large degree can govern, in many cases, the shape that the cell takes on. And so we've been interested in ways to measure properties of the cortex. And what we published just last spring is a way to do this by actually creating an acoustic standing wave inside this diving board-like structure. It turns out if we vibrate it at a certain frequency, the same one that we use to measure mass, there's a part in the channel where when the cell passes, it's not sensitive to its mass, but it's sensitive to a mechanical property. And what happens is that these acoustic waves in the fluid are actually squeezing the cell by a small amount, like 10 nanometers or so. That squeezing results in an energy loss that actually changes the mechanical frequency of this device. And so we could read this out in a way that's very non-invasive to the cell. And it's also just as fast and simple to get as our mass signal. So as that cell flows through, actually we get two signals. One part tells us the mass, and another part tells us about this kind of acoustic scattering cortex aspect of it. And so I'll show you, this is what we're able to do. So this is an example. So we wanted to look at mitosis. We thought that would be a nice place to apply this to just to see what it can resolve. And so what I'm showing here is traces from 24 cells that have been averaged together just during mitosis. So the scale here, oh geez, I'm just still not used to the pointing aspect here. Time after anaphase. And so we've aligned all these up because we have cell cycle readout as well. And so you can see these changes in the stiffness in the acoustic scattering that actually, when we stain for actin, we can relate this to what's happening in the cortex. So in the first part here, the cell's actually swelling and the cortex is thinning out. And then in the second kind of dynamic structure here, this is actually really during cytokinesis where there's a partitioning and there's a concentration of more actin closer to the furrow compared to the poles that are changing kind of the overall stiffness of the cell. So we know this readout can be very sensitive for looking at changes that happen in the cortex. So with David, we in his lab and others at the Farber that David works with, we now are starting to look at clinical samples, and we've done a few with the team that we have on this now. I'll show you just one example. So these are patient samples. These are mantle cell lymphoma patient samples. They're frozen, they're fresh frozen, and we thawed, and we now have measured them. And so this is from one patient where what you're seeing here is the normal lymphocytes that were sorted out. And you can see there's, in terms of mass, the stiffness kind of, there's some heterogeneity across that, but there doesn't seem to be a big dependence on the mass of the cell. But if we look at the tumor fraction from that same sample, you can see a distinct difference. So there's a clear subpopulation, if we go back, that's not there in the normal lymphocytes, but when we look at the tumor cell fraction, we see. And so we're excited to see this sort of structure in the data that we can go after. And so what we're really interested now is what are those cells? What do they look like? We'd like to sequence them. We'd like to understand what governs the heterogeneity and stiffness. What are the genes that describe that variation? And so a lot of effort now in our program has been focused on how do we get those cells. And so what we're setting up is a way to take the SMR, or mechanical resonating device chip, and connect it to a separate sorting chip that would be very fast at a way to be able to bin sort these cells based in kind of in real time, something that a flow cytometer has done for a long time, but we have to really build this custom to the requirements of our chip. And so this is actually being built as we speak. In terms of like what our throughput could be, so in the last year, we've been able to have a 20-fold increase in how fast we can measure cells. And we do this by having a parallel array of these sensors. So the cells can go through any one of these, and it just directly increases our throughput. And so we're about 7,000 cells a minute. And in this paper, we also have developed a temporal deconvolution approach. It's kind of like spatial deconvolution from microscopy, but the analog in a time domain signal. And that has allowed us to go up to about 24,000. Well, we haven't done this yet, but in the next generation devices, we are projecting 24,000 cells a minute. So in a couple hours in a morning, we should be able to go through several million cells in this way. And the challenge will, of course, be to sort them. But if we see these properties, we should have a way to be able to collect them. So let me stop there and just acknowledge the team. It's just been a really wonderful collaboration between several different places at Farber and actually at 2 different labs at MIT. And we're very grateful to have the Allen Foundation funding for this. So thank you.
We have time for a question.
Many thank yous for an interesting talk. I'm wondering if you can spend a minute talking a little bit about the relative advantage of doing this versus sorting with a flow cytometer. You can do size, granularity, and get high throughput. What advantages do you see here?
Okay, so I actually made one. I thought this question might come up, so I actually have a slide for this. So David asked me the same question quite a bit, actually. So we've done comparisons to flow cytometry. So this is one example. I'll show you. We have other ones coming, but so this is simple. These are just polystyrene beads, 6, 7, 8, 9, 10 microns. When we flow them through our device, of course, we see really clear mass-type peaks. But when you do it, so this is what we have in our core facility, BD machine. Here's forward, here's side. So look at this carefully. So you can actually see, not only are these things broader, but qualitatively they're different, right? Here's a 7 micron peak first, and then there's a 6. The green and the blue are also switched up there. And actually Mark Kirshner's lab has done work in this area as well. And the bottom line is that scattering and flow doesn't tell you mass. It tells you something else about the cell, but not its mass. And so I think that's really the main difference, is that we are measuring something very directly and we're measuring it very precisely. And so that slide, I think, reflects that.
Yeah, and there's one other aspect that we just didn't have time to go into, and that's that we've utilized this same approach to measure responses to therapeutic agents in very short time course. So we see very rapid changes in the accumulation of mass in growing cells and even loss of mass when put in the presence of an active agent. So we can use this as a phenotypic screen in a way that flow cytometry is simply not amenable.
And maybe one last thing, we could take very low input samples as well. That would be hard to do by flow.
I was just wondering whether these measurements are very expensive. Another way of saying it is, imagine we had 1,000 blood samples from the Framingham heart study. Could you profile them this way? Or?
Yeah. So I don't think cost will be a problem. The machine itself, the machines that we have in our lab cost about $10,000 to $15,000 to build. The devices are very expensive to make, but when you make them, you make thousands of them at once that can last us for like 7 years. So I think that's pretty, so it's just in terms of the throughput. And I think a lot of these samples can be run at time scales of minutes. Depending on how many cells you need to cover, it depends really what question you're asking, but minutes to 20 minutes per sample. So I think you can, and we have, in the company, we have 24 of them that run in a CLIA lab. So I think these are not surmountable.
And with the grad students, we found as long as you supply pizza, you can really get an enormous amount.
Yeah. All right.
Okay, thank you so much.